One project in SC Human, GSE75140, includes single-cell profiling of cortex cells. In the source paper, authors used a set of marker genes for cells within the developing cortex, including apical and basal progenitors, as well as neurons at multiple stages of development. We can search for these genes to recapitulate this figure, which allows you to explore the data directly. Since we're interested in data from GSE75140, let's pre-filter for this project. This project profiled multiple tissues. Let's also filter for CNS. Now let's search for the marker genes. The default view for multi-gene searches is an expression heat map, normalized by robust center scale, that is, by centering values for each gene, then scaling by median absolute deviation. The heat map is pre-filtered based on the sample filtering in the main LAN tab. So, only CNS samples from GSE75140 are being shown along the x-axis. The genes we search for are on the y-axis. Let's recreate the figure from the paper. First, flip the XY axis. Now click Change Chart Properties so we can rotate the labels on the X and Y axis. Let's also hide the dendrogram to use the original order of the genes as searched. Next, let's change the normalization to no normalization. Now we can specify a custom scale for the heat map. On the legend, right-click and choose Change Color. Let's use a TPM of 0 as a low value, 6 for a middle value, and 12 for a high value. You may have to experiment a little to get it right. Finally, let's change the heat map colors. Hide the color bars. And adjust the width. And see how well it matches.